Well, you could see it online, see if it's still herky-jerky.
With the zoom here, I'll take a look at it and see. Matt and I troubleshot it for a while to no avail.
Yeah, you can open it in the web browser back there.
I know, I can't remember the URL though.
I've got it.
That's what bookmarks are out for, you know? I don't know why. 122 maybe?
212?
It's 10.1.70.122. I think.
In fact, it takes me to Robert Waters' pirated music.
Napster lives.
212. You're close.
212. Okay.
It's still jerky on the video display site. So
okay.
And
interesting, because on the
yes, I know
the web, it's not.
Yeah.
What?
It's a network issue. The camera has stored up a jillion stills and is now downloading them, and it's maxing out the network connection.
Yeah.
That's the operative.
So yeah, so that's part of the upgrade that we're looking to do one of these days soon. Is it just has a gigabit connection, and we're going to go 10 gig.
Nice.
And dedicate. Well, there'll still be the camera itself still 1 gig, but it's, you know, there's other stuff riding along with it.
Is the is the vehicle connected to the network when it's on deck? Yeah.
So so one of the channels on the fiber optic is the gigabit Ethernet. There's a media converter.
So
but it's shared amongst a few different things, you know.
Bridge nav.
But when the vehicle's on the deck, is it powered?
It's all still connected the same way.
Okay, so you can be downloading them when you're not in the water.
Yep.
Kind of thing. Yeah. Okay.
But the caveat being the camera gets too hot if you leave it on for a long time.
30 meters at parallel 0,
especially out here, huh?
Yeah. I mean, I'm not up on what the NDI does, but I know it gets hot.
Mm-hmm.
But on the deck, you don't have to have NDI on to do file transfers. So it'll go a while longer with just the camera only.
Yeah.
So I'm told.
So we do have temperature monitoring on there. So if somebody's kind of watching it, then. So right now it's 64 degrees in the camera.
Wow.
Yeah.
And the water temperature is
2.
Holy cow.
Yeah. Well, so it's a titanium housing and it doesn't conduct heat very well.
Uh-huh.
Yeah.
But it is the NDI transmitter that.
Makes the NDI is on, so it gets cranking.
Yeah.
So
a lot of that heat.
you're saying the titanium is not a great conductor?
No, it's not.
But it's not a bad conductor, is it? Like it's not like an insulator.
Not like aluminum. It's not great.
Huh.
And so a part of it's that the NDI guts are not like in contact with the housing.
Oh, right, yeah.
So it's got a little fan that blows the heat around in there.
Oh, so air is an even worse conductor, isn't it?
No. It's just you're trying to get the heat from the air into the titanium housing.
Yeah.
Yeah. So I haven't done the math on that, but
you can borrow my slide rule.
Wait, I thought we were going back to abacus.
Abacus, you borrow my abacus.
No, that's definitely a ChatGPT
Robert,
thing.
you're pulling.
Sorry.
Bono, any comments in the chat or questions?
There's a question coming in for...
Faraday, alive.
Oh, what
Alive-ish.
is it?
Ish. It looks way better than any of the others we've seen.
All right,
It's
zoom in.
upright.
Let me put a highlight on this.
Looks like someone needs to change their filter. Feeder.
So definitely not as big as the one that we saw down on its side, but
That is sort of a DNA pump thing there, right?
Yeah, so I feel like, I'm not sure what's on it, but like, is that sediment or is it organic, the brown?
it might be its color
Ooh,
too. I
what
mean, I
is
feel
that-
like all the ones
What is
we
that little
saw
blue
before.
thing in there? There's like, oh, I was just seeing through it. There's holes in it.
Holes.
Yeah, you can see through it.
Okay,
I feel
prove
like the
that
ones
water's blue again. Nice.
Nice job. I feel like the ones we saw before were also kind of a yellow
Bridge
color,
now.
so I'm not sure if this is sediment or natural pigmentation.
Can we move 30 meters to the north, please?
I guess all the pictures I've seen are,
Thank you.
they're white.
You want to get closer?
Nope.
We're sort of waiting around
Oh.
for the ship to catch up.
Let's see. We need a sample? I don't
We're
know.
We're not that far ahead.
If we
Don't get
stopped
crazy
here.
now.
Oh, someone said we're sitting around here. Can you swing around to look at the other side of it?
Oh,
Aha.
Oh.
there's
now we know
so much
its
more
secrets.
to it.
Now we know your
Yes.
secrets.
That wasn't on my bingo card.
Wow, it's like a big basket.
Like it looked like a chair.
Oh,
The
wow.
Hollywood Bowl.
Everyone sees something different.
Oh, now we really see those formations we saw in that big tree, like one with those kind of
You want to zoom in?
fingers coming up. Wow,
Wow.
that is really something.
So all those larger passageways lead to smaller holes, and either Venturi or Bernoulli is having a field day.
Yeah, I looked it up, and Bernoulli would be for viscous flow. Venturi's more for air.
Ah, nice.
When you have a change in pressure, that changes the flow.
So as a lava guy, I'm more Bernoulli, high viscosity, low Reynolds number.
Always about that laminar flow, right?
Yeah. Not a huge fan of turbulence. Pretty hard to describe mathematically.
You want to look around a bit? All right, that's it. We gotta go.
Can you pull back a little bit for the cinema cam to get a good shot of this whole thing?
All right. You're gonna have to tell me what.
Yeah,
looks
you're-
good. Is that
Down a little lower.
Oh.
And to the port a little bit. Oh, never mind. I got some pictures already.
what?
Put the thing back up on the... Oh, yeah, you're good. Now back a little bit.
Work it.
All right, all right. You're a
There you
sponge,
go.
show it to me. All right, we'll call that good.
Good job.
Yeah, check out Sponge Illustrated next month when we, whatever happened to magazines? Do they still exist?
Really.
Only in airports.
Yeah, seriously. That's probably printed in each airport. They have like a printer. Like, oh,
A
make
single
another
printer.
magazine.
Stopping pump 3.
Bridge nav. Can we have another 30 meter move to the north, please? Thank you.
And starting pump
Gonna
4.
reset the DVL.
Sorry. How many liters did you do for pump 3?
It was 153. I got distracted by the sponge.
It's been a remarkably consistent kind of community all through this dive.
That's sample 216.
Thank you.
Set another prim know-it off in the bottom right-hand corner.
Be the
Yep.
camera guy. That's wrong.
I don't think it's anything new, just confirming.
Zoom in, Dave.
Yep, same thing we've been seeing.
No associates in that one.
Nope.
Oh.
It's always fun when we zoom in though, you can see little sponges growing, some of the little sea stars and things in the background.
All right.
Thank you.
So we are once again up on top of the ridge and not having current here. That's pretty cool.
much, yeah.
Johan, when we finish this move, before you call in the next one, there's a little happy shrimp.
Mm-hmm.
Might want to look for a rock sample location.
All right.
Looked like that shrimp had auto XY on. Just sticking in one spot. Oh, no, it failed.
Well, I think the current's just more than he can handle. It can handle. Zoom in, Dave.
looks like that one has some good paddles.
Yeah.
Doing some parkour, running off the wall.
Upside down.
Ready for its close-up.
Oh my gosh, what's that, all that stuff hanging down?
Eggs.
Eggs? Look
Oh,
like
okay.
it.
Oh. Yeah, maybe.
Yeah.
Sorry.
Oh, that's good. That was cool.
That's cool.
Is that a fish or another shrimp there to the left?
That's another shrimp.
Yep.
Up on the wide camera as well.
Okay.
All right, can you center it back up again?
That larger white thing in the fisheye, a sponge or a coral coming up?
Oh my goodness. I think that's a healthy Faraday. A little dangerous to make the guess from here, but yes.
Oh, there we go.
Look at
Okay.
that. Oh, what a beauty.
Oh, that's lovely. How tall do we think that is?
We have lasers.
A meter and a half, 2 meters.
They do look a little skeletal, especially when they're white like this. Almost like a backbone.
So I'd say this is the first one we've seen that's in a very healthy state.
Yeah, this is gorgeous.
Which makes me think that, I don't know what that brown stuff is.
I was wondering if you could get a little piece of that one.
Probably.
We are all stop. Oh,
Ship is stopped, yeah.
oh, okay.
So I think we can.
Got you.
Probably best approach is to use the slurp to kind of like break off a little bit of it.
All right, we might be a
I
little.
think one thing that we did this morning that Tito did that worked is get a little piece with the manip and then bring it to the mouth of the slurp and then turn it
so
on.
snip and slurp.
Snip and slurp. Do you want to look at it from the other side to see if that's easier to snip and slurp, or?
I'm okay. I'm just too close to it. I can back off a little bit.
Okay.
Is all. You could probably just, yeah. Esteban, can you push the basket or the porch out?
Yep.
Sorry. Porch out.
Bridge now.
Pump 4 is paused.
Can we
good.
move
That's
10
good.
meters to the south, please? Thank you.
Okay, we doing snip and slurp or are we doing slurp?
Snip and slurp.
You zoom way in, Dave. I'm too far away. Yeah, I need to get a little closer.
I bet on the other side of it, like that one we saw before, it probably sticks out.
Oh, yeah, that might be better. We're on the
Convex
yeah.
side. Yeah.
All right, we will try the other side. Can you tilt down some so I can see the front of the basket? Still staying in place? Are you gonna tell me right when I'm all set up that we gotta go?
He's coming back towards you, I think. Is that right, Johan?
Yeah, I moved a little bit closer.
Yeah, this is better.
Oh, look at that.
Can you, we're all right. We're too up close and personal now. How about that? Right there. Okay, tilt up.
Yep.
Can you zoom in more? Uh-oh. Why is that joint not going? Because it's locked.
Ah.
Can you tilt down some, Esteban?
Yep.
Does it look okay?
Yep.
Yep.
Perfect.
Okay. You get the camera in and standing by for suction pump.
Better check what jar
Yeah,
you're
you
on
gotta
there.
get on the jar and all that good stuff.
Yep. Just get the camera.
And then can we zoom way in?
What?
3
Which
is
ones do you have
open.
available? Three's available?
Three's available.
Copy. Okay. Suction pump is on.
Can you center that up anymore?
Let me see if I can pan right a little bit more. No, that's about as much. Let me tilt
Can you go down?
Yep. Okay. Suction pump is on.
Yep.
There you go. Almost in.
Yeah.
Yeah.
Nice.
Swish.
Swish.
Yeah, let it settle.
Yeah.
Great sampling.
All right. You want to rotate that to the flush and
Yep.
give it a flush?
Before you lift off, see if we got any rocks that are grabbable around here.
Yeah, we might find really good ones.
Okay. Flushing.
It's like you got it all.
Yeah.
All right. I don't see anything loose around here.
Well, we'll zoom out. Maybe there is.
You could there, suck the porch in.
Yep.
Oops. That's not a thing.
There you go.
I'll come
There's,
up a little bit.
That thing right
Suck
there.
it in.
Yep.
Does look pretty glued down.
Yeah. You want to re-examine the sponge?
Yeah, that's beautiful.
You zoom in there, Dave.
Oh, there's a little
Maybe a squat lobster?
squat lobster in there. Oh man, the cinema cam has a beautiful shot right now.
Do you want the lasers off for that shot, Adam? Or better to have
sure.
them on?
No, we have a bunch with it on, so can you secure the lasers for a moment?
Laser secured.
Is this kind of sponge same as the one that we saw earlier?
Yeah,
Yes.
very a day.
A
Oh, look at
different
that cute little guy.
color.
That little thing. That's hard.
A little
yeah,
lobster.
it's really hard.
It's
It
a little
is.
squat lobster.
It is like those circuits are really
Really
well
hard wired.
established.
Yeah. Okay, I'm deeming this the most beautiful sponge we've seen on this trip.
I can go with that.
All right, I'm happy on the photo front.
Okay.
That's a highlight comment for ROV pilots.
Yeah.
Quote, you ROV pilots are wizards with the arm and no 3D perspective. Well done. Bob
Look at this
Natty.
shot here with the bolosoma and the Faraday. They're all kind of oriented into this, must be the current flow,
Yeah.
trying to catch particles. Really beautiful. Just great example of all these different sponge morphologies.
Oh, and there's a big dead one, yeah.
Oh, yeah.
Circle of life.
Circle of life. I actually don't know how that tune goes. But I'm willing to try random tunes until I get it, okay? Erica, comment coming in on the chat if they would like you to sing the Circle of Life. Is that okay?
I don't even know how it goes.
Hard no on that one.
That was amazing. You're like, okay. La, 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 la. Nope, I don't know it.
I'm not seeing any rocks that are snaggable.
All right, we can keep going. We'll keep our eyes peeled.
Adam, you need to bring a jackhammer for the next time you're out here.
Well, we discussed that. It doesn't work.
Doesn't work?
Doesn't work. You gotta have every reaction. There's a
Equal and opposite reaction.
Yeah.
Yeah, yeah.
Well, if I bring also the harpoon anchors.
yeah. Yeah.
Well, there was were you involved, Bob, when they had that gripper
Yeah,
drill
the
thing?
JPL,
Yeah.
the alien head gripper.
Yeah.
Yeah. That was quite the contraption.
Okay, these look kind of grabbable.
Yeah.
Oh, let's go for it.
Let's do it.
Or it's, from a distance kind of thing.
Oh, I know.
Yeah, correct.
Huh?
Well, there's
I think
Yeah.
that one looks like it's just sitting there, yeah.
they're just,
Until you get down here, and then you're like,
Yeah.
You know, if you want a little bit of pork chowder, is that okay?
we're all right.
Okay, cool.
It's worth a shot. Or not.
Wow.
Yeah, pretty in place.
I love how we all get silent, just waiting to see if there's any movement on any of these rocks.
What about that there? Can you get a grip on that? It's just a flat piece, huh? Yep. Nope.
Yeah.
All right,
I'm
we'll
not
move on
budging.
and see what else we see. Nope. Nope.
It's so different from the rocks at Vailaluu that were just
You look at them and they fall apart.
breaking off with the slightest touch. Oh. And is that really just a factor of the manganese crust on these?
Yeah,
Like this
yeah, it really is. It's like
just really strengthens all the lava.
yeah, I mean the lava, all the
Formation.
parts that were really friable and fragile on this seamount were gone, 100,000 years after it erupted, and then what's left behind is the solid parts, and then it got glued together.
Are we going to be moving for a while?
I don't know. Bob, are you still looking out for
Yeah,
loose
I'm
rocks?
just kind of poking about here.
Hold on to restarting the pump then for a little bit.
Yeah, I think that's probably
I'll
smart.
try and not bounce off the rocks much.
Adam, do you have a favorite song for rock hunting?
Nope.
No.
I gave you a suggestion.
It's got to be a Rolling Stones thing, right?
I want a rock. I want a rock. But I can't remember who sang that. See, the ones that are out in
Isn't that
front
Van
of us.
Halen?
No, I think
No?
it was a different hair band.
For those about to rock, we
Was
salute
it what was Dee Snider a part of?
you. Twisted Sister.
Twisted Sister. Could that have been it?
No, I
Okay.
don't think so.
Ooh, that's a different sponge. That is...
Gonna reset the DVL.
Well, Val, I'll give this one to you.
You might lose your lock again anyway.
It is different, isn't it? It's more rounded than the other ones.
Yes.
Bigger, bigger pores there as
And
well.
Connected to the
But
bottom,
it
right?
does look like it still has some kind of hairy projections holding it onto
zoom in,
the
Dave?
bottom. And of course I put away my sponge ID section. So now I got to pull it back out.
Wow, that's little extra filters in there.
Yeah, look at that. It just amazes me how fine these structures can be inside of these sponges.
So maybe in the Poliopogon family, but let's see. Asako, what do you think of this this sponge here? Other than it's very pretty.
Something weird going on with your tether action there. All right, you seen enough?
Yeah, I've seen enough. Keep our eyes open for more of those.
You got to move on?
No. Are we moving away?
Let's move on, yeah.
Okay. Bridge nav.
There's a Gorge up above.
Hi, can we have a ship move 30 meters at bearing 015, please?
You tilt down a little bit.
Thank you.
It might have just been a different type of Poliopogon. I see a couple images in here that look kind of similar.
Oh, darn.
There ain't nothing budging.
Yeah.
Maybe we need to blast some Twisted Sister at it. How much force does it take to break this kind of crust, Adam?
Slightly more than a craft manipulator can apply.
Obviously.
Yeah, it's actually a really hard question to answer, and one I've kind of thought about, because one of the things that controls how lava moves across the seafloor, across the land, is the viscosity of the fluid, but the other thing is this solid crust that forms on top that produces an effective viscosity for the mixture of the crust and the liquid. But it's hard to know what the material properties of that crust are, because it's not necessarily continuous, and you know, it's fractured, and that makes it weak, but underneath it there's a kind of very viscous layer. I don't know. Answer is I don't know, but it's a very interesting question.
Okay, thanks. Okay, restarting pump 4.
Oh, we're starting to get more sponge diversity.
Yeah, that's a pretty one on the upper left, just
Yeah.
off camera now.
Can you zoom in, Dave?
That one's got like little ribs
is that
going
one?
across it. Oh, and that's
It's one of the closed-in ones we were talking
Yeah.
about. There could be shrimp trapped inside.
Is that them at the top?
Or making the choice to permanently join together as a partnership.
Can you look down the throat? You get
Oh, that
more
might be
zoom
them right
Dan?
there at the top, maybe?
That looks like it's on the outside.
Yeah. Again, just looking at these structures, so amazing. And I have seen a similar, I've been able to hold a similar sponge, and it's just fascinating to me.
Yeah, like how
They're
does
so
it
hard.
how does
Ready
it
to tilt down.
Yeah, try
biologically
and look down the throat.
decide to make that netting? You know, like where does it branch off?
Can we move 30 at 030, please? Thank you.
I think that shrimp is inside. I see glowy eyes. And it's moving.
Okay, yeah.
Yeah.
Oh, there's its partner.
So is it the Japanese wedding sponge?
I don't know what that one. Let me take a look. Okay, not sure what happens when you Google Japanese wedding sponge, so we're gonna find out.
You find out.
Yeah,
Maybe just don't read off all the comments.
it's a euplectella, and that's what this is.
I think that one might have, it looks like there's little balls inside. Could be eggs inside the shrimp.
Wow, that's.
Or maybe not. I can't focus
can't
my
see it
eyes
that
well enough.
Yeah. All right.
Thank you. That was great.
Yeah. All right.
So Osco suggested, well, it is a euplectellid and dictyaulus as the species, perhaps.
It looks like another one right up here.
Yeah.
Zoom in, Dave.
That one seems a little more rounded at the top.
Yeah, that looks a little different, perhaps.
Little bulbous.
Maybe if we see it at top-down view.
And what is that right behind? Is that a stalked crinoid or coral to the right?
To the right.
Or two? We've lost it.
Okay. No, that's gonna.
It's okay. We don't
hold
have
its
to go back.
secrets.
We'll find more. Oh,
Bridge nav.
Can't look down it.
Yeah, don't worry about it. Oh, I see. Yeah, I see your
Can we
stalked
move
crinoid
30
there.
meters at bearing 020?
Very
please?
delicate.
yeah.
Thank you.
I think that might be the first one I've seen on this dive.
Yeah.
Okay.
Thank you.
Oh, there's a big polyopogon.
Mm-hmm. A shrimp.
Or a little mama shrimp. Wait, if a shrimp has eggs, does that mean it's female?
Yes.
Okay.
And that's one of the ones you can actually tell the sex. A lot of these other critters, no idea.
No way to know. Or both.
Or they might be both.
Yeah.
A lot of them are both. The sponges, for example, hermaphrodites, which means that they produce both eggs and sperm.
You zoom in, Dave?
I saw that too. It was-
It's just a base.
I think it's, yeah, a sponge base.
Yeah.
Looks like there might be some little corals or something to the left, though.
Yeah.
Might be a little
tiny.
stylaster or
Little tiny
something.
ones.
Itty
Yeah.
bitty.
Those might not be corals though, huh?
They might be hydroids.
Yeah.
Yeah.
Right.
I sometimes will sit for an hour on a shallow reef in one spot just looking at all of the little itty bitty teeny tiny things. I wonder if you could do the same thing here in the deep sea, like if you really settled down
Oh yeah, for sure.
and just sat there, how many little things you might be able to find.
And like, if you were kind of oriented towards discovering new species, that's what I would do, because you know, no one's collecting the.
Itty bitty things. Yeah. But it's so hard. We have such limited time down here on the bottom, and we've explored so
Bridge
little.
down.
Is that a fish
No.
left of center?
Oh,
You
left
zoom
of
in, Dave?
center.
Left
I
Top
was looking
left
on the
corner?
right.
It is a fish.
Oh,
It's
yeah.
a fish.
Can we move 30 at 030,
It's running away from
please?
us.
Thank you.
Maybe a halosaur?
Yeah, that's the one I door dashed so that Val would not be disappointed with the dive.
We need a fish for Val, please.
I
One
actually
fish.
saw the same one, but much bigger when you were explaining eDNA.
What?
Yeah, it was on the fisheye cam.
Oh, you should have shouted out.
Fish!
Exactly. Full stop for
Fish
fish.
on, fish on.
It's very bashful. It just keeps hiding from us, doesn't it?
All right.
Thank you.
Yeah. Yeah, center me up. I don't know where I am. And tilt down some more.
Erica, don't hesitate to shout out fish in the future.
Okay.
Or other cool things. Feel free to jump in here and add on. We can use all the eyes we can get. Ooh, what is that red thing towards the center? Is that an anemone?
I think you're right.
There.
Might be our first anemone that we've noticed.
Yep.
There we
that's a
go.
Beauty.
Oh, it might be a little snail at the base of it.
Oh my goodness.
or rather large snail actually, with
Yeah.
a long spiral shell. Could be a hermit crab too, but
No, I think that's a snail, and a lot of snails, don't they eat these things?
possibly.
I bet.
Is this another predation event for us?
Maybe, but I know that folks are interested in sampling those things. Did we sample that snail?
How you want to get it?
I think it's probably a slurp, yeah.
Straight slurp? Yeah.
Straight slurp.
All right. I'll stop.
Bridge nav.
I'm gonna stop pump 4.
Can we cancel our current move and
Can
move
you push the basket out a little bit? Or
south
the porch? Sorry.
at a bearing of 210?
Oh,
30
that's
meters.
good.
Sorry, 30 meters at 210. 210.
How many liters did you go for that
Thank
one?
you.
I'm just pausing it for
Oh,
right
you
now.
still
I'm just
down
gonna
so I
keep
can see
going.
the porch more.
Thank you for checking.
That's good. And set up on the
You should say hi back.
whatever canister we're on.
Hi, Megan. Thanks for tuning in our chat box questions on our website.
Um, 4 is open if you want.
Copy.
Ooh.
There we go.
You tilt up and zoom.
There you go. See it.
Oh.
Wait to see if it comes in. I'll give it a jiggle.
I think I saw it go
There it
in.
is.
There we go.
And that may be a Cerithidae
Settles.
type of gastropod.
There we go.
Hey.
That's
Oh,
218, sorry.
No,
looks like a
thank you.
show.
Yep, little snail inside.
Well,
That anemone's probably like, thanks. It was eating my toe.
He is sucking the porch.
Yep.
I gotta hurry up.
Yep. Bridge nav. Hi Flavio, can you cancel this move now and put in a new ship move of 30 at 030? Thank you.
I love it when the numbers line up. 30 at 030. That's awesome.
Me too.
Why is that so satisfying?
Alliteration.
I guess so.
Yeah.
Symmetry.
Yeah.
We love symmetry.
Oh, dang. Don't these rocks look
They
grabbable?
look all grabbable except when you get up close.
Oh. No, I swear they are.
They do look very grabbable.
If you want to look,
say.
I don't think we're moving for a little bit.
That one's just all by itself right there. Isn't it? Those don't look ungrabbable.
No, they're, you can get the jaws around them.
Right?
Yeah, we're going to get one of those.
Do you want to do it on the fly, or do you want me
Yeah,
to
I'm going as quick as I can.
copy?
I have a question coming in about the slurping of the snail from the anemone.
Yep.
How much of an anemone would a small snail like that eat?
Oh. I don't know. Yes.
All
That's how you do
right.
it.
Bravo.
Forward box just to be quicker, I suppose.
Nice.
All right. Box out. Yeah, you got scoopies in there.
You just drop it in lambda. I think it'll fit in that little front part there in front of the... Oh, maybe. Maybe it's pretty big.
Yeah.
Oh, never mind.
Jumbo rock.
Side basket, starboard basket. You can pull off the bottom if you want, if you're getting...
You
trying
might
to
have
be
time.
rapid here. Can we switch over to...
Yep.
On the side.
Okay. Go out. Which one?
They're all open except C.
I think this is probably too big for... It's gonna
Yeah,
have to
put
go
in a
around.
big boy, yeah. E or F, there you
F.
go.
Okay. That's sample 219.
It's the Steph Curry of rock putting away there.
Yep.
What time is it? 22.
Cool.
Is that our first rock for this dive?
First rock of the dive.
Oh, awesome.
And you know, our tradition, once we get our first rock, Erica sings a song for us. So here we go.
Erica, your mic's muted.
That was very
I
fast.
couldn't hear you.
She sings it in her head, though, so sometimes you can't hear it.
Okay.
Meet at the social deck after lunch.
Which one, the karaoke deck or the...
Of course, the karaoke deck.
It would be awesome. Have we ever had a karaoke machine on this
Yes.
ship? That must be great.
That's not the word I was going to use. And just noting, we've passed
like
a
we're
few
coming
sea
up some.
cucumbers.
Ooh.
So they're a little bit paler than the ones we saw before. Also some chrysogorgias while we were flying over. And I'm going to restart pump 4 now that we're moving again.
We also had a giant Mario Kart competition
Ooh, I don't think I'd be very good at
on
that.
a mapping leg.
Yeah, there's some good Mario Kart players.
Yeah.
I bet.
Everybody,
Yeah.
a bunch of guys brought their controllers, and so there were, custom controllers.
Oh, really?
Lots of, there was a tournament, lots of strategy.
Oh, there's a nice coral.
Esteban, will you start
Zoom
coming up?
in, Dave.
Either Primnoid or Rumellagorgia
Bridge down.
with some stars and crinoids. That's pretty.
Can we move 30 meters at bearing 015, please?
All right,
Thank you.
moving ahead.
Oh, this is a good question. Where and how are the biologic samples processed after collection?
Where?
Yeah, where
And how?
Yeah.
Yeah, so actually if you tune in to the live stream after the dive,
Yep.
you can see the team doing the sample processing. There's a laboratory on the ship where the samples are photographed and preserved in various ways. So oftentimes
Zoom in on that, Dave.
in ethanol or in, I don't think we're doing any formaldehyde on this, but sometimes...
New sponge on old sponge.
Oh, yeah.
I just say Erica's been helping to process all those
Oh yeah, why
samples.
don't you tell us what you do, Erica?
So we take photos, split up samples. I know they go to different agencies and universities. And then we preserve it in ethanol. And if not, then they just go in frozen. Or Adam's rocks just sit on the table.
Yeah. Mine are happy to just dry out. Otherwise they get stinky after a while.
It is quite the rock collection down there. We were talking about possibly stacking them up in Jacob's room so he could get really
Get in tune with them.
get in tune with them for his PhD
friends.
research. We were thinking we could maybe put some googly eyes on them.
Sure.
Really,
You know,
make
we're going to
them-
do some rock cutting this afternoon.
Yep.
Anyone who hasn't had a chance to cut a rock.
Looks like we're coming up.
Come on up. Moving ahead.
Oh.
Oh my goodness, we're already coming up on shift change, aren't we?
Yeah, it's just a few minutes.
Almost noon here,
What we
goodness.
got there?
So,
Oh, that looks like a
zoom in.
different glass sponge maybe. With some little shrimp inside, maybe? See two little specks.
Yeah,
It might be on the outside though.
I think I came up with an idea on this before, but I can't remember what it is.
Did that make it in the cinema cam?
Yeah, it's like lined right up. It's perfect.
We're getting sediment.
Yeah,
You
Little
got-
shrimp
free to go,
there.
free to go.
You want to?
Oh, gonna pause the pump.
Up, up, up. Yeah, that's good. Try and get the white bits in them. Yeah, that looks like, yeah, that's shrimp.
Oh yeah, it's moving.
Hey. All right, I'm out.
You can see those really fine hairs that it uses to catch detritus too on the sponge sticking out, just thread thin.
Atalanta's still moving, yeah. So we're going to want to come up.
And you might hear a little pause as we switch watch positions.
Aha.
I have been. I love it. It's just taken some practice. Yeah, I'm not used to it, but I really like it whenever I think of it. Yeah, why don't you come up? I'm moving as fast as I can.
The ship is now stopped, but
Yep.
it is still.
I'm restarting pump
Coming
4.
along.
That's better.
That looks
That's
a lot
much better.
Yeah, fun watch change. Oh, it's been great. Mm-hmm.
Levels of understatement are just classic. In the place where I hit things with the vehicles.
I'll tell you that is not our closest of the shift.
Yeah. They've been having some close moments. Like Rennie can feel them when he's like in his cabin. Oh, good. She came up there. She intervened. What's that? Yeah. I'm real concerned. Concerned dad. He went fully concerned.
Hello. Okay. I don't think we've seen that yellow coral to the center there before, so it
Are
might
we
be
gonna? Is
worthwhile.
there a risk of cutting the dive short if we're done, or is there just like lots of cruising around on the surface?
There's lots
Okay.
to do along the top of the
They
hill.
wanna do a
once we
whole
get
what?
up there.
Okay.
Yeah, if Megan zooms out, just getting to the top of the hill isn't enough. It's driving along the length of the top of the hill, so I just checked in with Adam. He figured it might be a recovery
heavy
on
again.
the 4 to 8.
Guess they've been picking up rocks. Oh, right, it was. Adam, you've reballasted the vehicle again.
It was only one rock, apparently.
It's a heavy one.
Yep.
It only takes one.
It
Yeah,
only
he
takes
was-
one.
He was thwarted many times, he said. It wasn't for the want of trying, but he also thinks we're just on the brink of tripping into a high biomass
Just
area.
on the brink of greatness.
Just on the brink of Meganness.
I mean, it looks pretty great as is.
Yeah.
Like, look at all these bolosoma.
But apparently that's why they're so far along, is because it hasn't been this great up till now, so...
Oh. Well, where we landed last night was fantastic.
Yeah. Well, actually, no, where we
Okay,
left them last
I'm
night was
ditching
on
away.
deck, but...
Looks like Atalanta is settling out, so I'm going to pull ahead just a little bit, and then I'm going to take this opportunity to ditch away.
Sounds great. Look at that, Bathy Pathy is your favorite.
Video? What's going on?
Just got in my chair just now.
Okay. Yeah.
There's black screen right now?
Yeah.
Oh, shoot, okay. Let's see what's going on. I also have black screen on Zeus.
So Val was just
One
asking me
moment.
if we could get maybe this yellow coral that's on the right. She said she didn't think we've seen that before.
Oh, the Bathy Pathies?
She said the rightmost one of the three
It
on
went
that
down
rock.
like the iris went down.
Yeah, that one's the Bathy Pathies.
So it might just
we'll
be
take
set
a
in
close
auto
look at
iris?
it. I don't know.
Okay, we're all good now.
Yeah. Science, if you'll give me, I don't know, 5 minutes to get things sort of sorted out, get reballasted.
Yep, we can
I
keep track
will.
of that coral while you're doing that.
Say
Make
again?
sure we keep an eye on that coral that we want to sample.
The Chrysogorgia right here?
No, the black coral.
Oh, okay, yeah. We can do that. Yeah, if we stay here, you'll sort of pass over the top of me, so just that. That's all.
Unless Megan, have we sampled this type before?
Yeah.
Okay, so maybe we don't need it in that case. I think Val was saying she didn't think we had sampled this type before. So if we have,
We
then-
got a shallower Bathy Pathies, but
It's a Bathypathes. Oh, we don't need this.
I think she just wanted to set you a kind of mean task for
Yeah,
the first
just
5 minutes.
You're awake now. Here's one the pilot can't do.
Is this, so do you guys want to sample this?
It sounds like we have them already, but Chris, you're
Okay.
What
You
depth
have
do we
5
them
minutes
from,
to
Mary? Yeah.
figure out
I
whether.
don't think it's a
Keep-
priority,
Okay.
but
Yeah, we have a Bathypathes sampled
And what depth
from
do we
earlier,
get
but
it,
not on this
get it from?
trip.
Oh, from a prior dive?
Yeah, from a
dip-
previous dive, not on this dive.
On this cruise or different
Yeah.
cruise? Okay.
On this cruise.
Megan, is there likely to be an issue of whether it was significantly deeper or not? For species
that one
and
is
range?
a different species, but we have had samples of these deeper species. I was going to look at the
Can you pan
branching
down for
pattern.
me? I'm gonna release a weight, so I
Okay.
just need to see the porch.
Maybe we'll
And you
scrutinize
can
it, Megan, and follow your judgment of
porch out a
whether
bit
it's
too.
new enough and different enough to be worth taking a snip. How's that?
Yeah.
Beauty. And pan down a little more. Great. Thank you.
Well, this depth range has been pretty well sampled.
Okay.
For these species.
So it's gonna have to be
and
something really
if
quite
Tina
special to
Molotova
merit.
was online, then I would say like leave it to her, but I'm not sure if this particular one warrants a sample.
Okay. Well, you've laid down the gauntlet for the colleagues online. And they can type now or forever hold their keyboards.
Yeah, is Asako online today?
She is. Yeah.
She usually
She-
knows what
yeah.
Tina wants.
Asako is right here and just said, hello, new watch team.
Hi, Asako. To get like situated with my my coverings. It's so cold in here.
Is it a good time to quickly go around the...
I think so, yeah.
All right. Talofa, Samoa, Lalangiatoa, Oingo, Isabel Halatuituia. Greetings and talofa, everyone. My name is Isabel Halatuituia. I am sitting in the Science Communication Fellow, and I'm pretty stoked and excited to be sitting here and checking out Malulusi. Where are we here at Malulusi Mount, right? Yeah, pretty cool. We were here early this morning. Unfortunately, we had to come back up, but we're at it again.
We were one of the pioneer species of this seamount.
Are we at
Because
Malulu Seamount?
Malulu.
Yeah.
Malulu.
Oh, sorry, I thought you said Vailulu.
No,
And I was like,
Malulu.
okay.
Malulu?
Yeah.
This is the one
Malulu
we
means
arrived at.
cold.
Yeah.
And it was. And
Yeah.
it will be again.
Even
It's
in
still
here.
cold.
Yeah, exactly. It's like 4 hours from now, we will remember why it's called Malulu Seamount.
Yep.
Yeah, so I'm Chris German, one of the Chris's on this watch. I'm here in the watch lead, so I don't actually do anything useful, unlike the other Chris, who's sitting to my right.
And hello, I'm the other Chris. And I'm in the data logger seat for this shift.
Hi, I'm Mary, and I'm also on the science team for this shift.
And what are you playing with today, Mary? Do you have both?
I have 2 Sages, so 2 dissolved gas sensors, one which measures CO2 and one which measures methane. And then I also have some eDNA pumps that are running for some of the other scientists to collect data about what species we have at different depths up this seamount.
So as you can gather, Mary's really, really busy, so that between us makes us a well-balanced scientist.
I am sort of put together now. So...
Let's zoom the Bathypathes.
Okay. Lasers on?
Lasers are on.
What's that?
The lasers are on.
No,
Oh.
do you want them on?
Yes, please.
Okay. Go for zoom?
Going for zoom.
In your front row, this is Megan sitting in the navigator's seat.
Gabby in the Herc seat.
Right in the Atalanta seat. And Samantha in the video seat.
So this is a coral?
Yeah, it's a type of black coral called Bathypathes.
Black coral.
And it's my nemesis.
Yeah, they're very floaty and wiry and challenging to continue to stay collected.
Okay,
I think
is
that's
that
good.
what you need?
Yeah.
Go wide.
So do we think that's something we already have enough of and we don't need to sample?
Yeah, I don't think we need to sample that one.
Okay. I did ask Asuko, but she didn't type either way yet.
Data. What have we collected in terms of biology
On this dive?
on this dive?
Well, let's see.
A rock.
Chris, you're confused.
No. That was Adam. That was Adam's take on biology. There was a rock. There may or may not have been anything attached to it.
What's this?
Okay, I'm going to get out front.
This black stuff, is that a dead coral in that crack between the rocks?
Oh, maybe dead sponge, hey?
Maybe dead sponge.
It's a dead sponge.
Okay.
We saw that on our first dive. There was all the
Yeah, I
really big dead
remember.
sponges. That's the same kind.
So I think we're at the top of like a little rise here, and that's why we have all these lovely sponges here.
So as far as biology collections, we've got 3 sponges and a snail.
And a snail.
Yep.
Ooh.
Oh, that's exciting.
Oh, the snails were really fun to collect. You know, remember like 3 lifetimes ago when we collected a bunch of snails?
Oh,
yeah.
With the suction?
And then they turned
Look
out
how
to be
you-
way bigger than we
Look
imagined.
how yellow
Oh, yeah.
those stalks are.
Yeah,
is
they're
that our
pretty
lighting or is that actually their true color?
That's their true color. They're lemon yellow.
Wow.
Yeah.
That's
Or
awesome.
like highlighter yellow.
It's like citronella or something.
Go for zoom video.
Going for zoom.
I'll just follow the base of this sponge up.
Okay.
So you
Yeah,
can
some of these colors can be used for like anti-predation. The chemical itself produces this yellow color. We're not exactly sure why these sponges have
You can go in even
this
further.
particular color. It's not really detrimental or useful to them, but usually when they have a coloration like this, it's because it's something that the sponge produces for another reason.
Megan, is this another Calophacus sponge?
This is a bolosoma.
Bolosoma.
Yeah.
Okay.
So you're not wrong in thinking it looks very similar. The way to tell the difference is where the stalk of the sponge meets with the head of the sponge and where the osculum is. So
Okay.
this sponge has like kind of a concave, like little cutout on the other side. So on the left side, if we pirouette around, you can kind of see it. And then it's got a little osculum, which is like a hole on top. So that makes this in the family Euplectellidae and the genus Bolosoma.
All right.
Whereas the Calophacus have the stalk that leads to the back of the head of the sponge. It doesn't have an osculum, and it looks like a little mushroom.
Ah, okay.
Yeah.
That's so cool.
I will try and remember that.
And Asuko has sent a message from shore saying that she wasn't sure about the Bathypathes either. She wasn't convinced we needed to sample it, but she's taken a picture and shared it with Tina
Fantastic.
so she can provide feedback.
Yeah, if
And
we
if we don't
see
get this
another
one,
one,
if we
Tina
see another
wants
one and
it,
Tina-
then we can
exactly.
grab it.
Yeah.
So what is our plan? Ooh, is that a Calophacus?
That one is a Calophacus.
Yes.
So now they're right next to each other. You can see the difference.
Uh-huh.
Bolosoma, Calophacus.
Yep, Bolosoma and Calophacus.
Woo!
It's amazing how you can memorize all of this names. I can't even pronounce or spell some of these.
Oh yeah, learning
I
how to
was
spell
gonna get
them all
stretched
was
out. I got
challenging.
sidetracked.
But what helps is if you keep typing them out.
Yes, I'm better at the spelling.
That's the
than
thing.
the
It's
names.
like if you lived in a culture with oral tradition, you might be a lot happier.
Yeah.
Or you could test out what Siri's spelling is like, Megan.
What spelling is like?
You could test out how good Siri's spelling is.
Oh,
You could
yeah.
just like you could dictate the names and see if Siri
I
can
don't
spell
know.
them correctly.
I don't know if Siri can spell some of these names. She gets really confused when I say some stuff.
But how long will it take for it to spell it? A while?
So we got a Resingid sea star, a Vermilligorgia, and a Chrysogorgia, or Bolosoma. There was a really interesting Chrysogorgia off to the left that I saw. I don't know if it's still in view. but it was tall, like a long thing stalk, kind of like a Iridogorgia, but we might have passed it.
Never go back.
Yeah, no, always forward, never back.
I have a question here. How long can the ROV stay underwater? I think the record was 3 days.
It was like
You're off
72
SPL.
hours.
Sorry, Gabby.
We may have gone longer than that. I can't tell you how long the record is. We've definitely done 72 hours this year, I think.
Yeah.
Maybe? But the ROV can stay down basically as long as it keeps all its fluids inside of it. It doesn't, its fluids aren't really consumables, but they do, like, the oils, like the hydraulic oil and the oil that surrounds all the electronics are, they're held in with seals, but there's always just a tiny bit of leakage, and we monitor those levels using pressure gauges and level sensors, and if the levels get too low, then we have to recover the vehicle. The other reason we might have to recover the vehicle...
Cucumber.
What's that?
Sea cucumber?
Oh, yeah. The other reason we might have to recover the vehicle, and this is much more frequent, is we just run out of sampling room. Like, we pack all the boxes with all the samples and all the suction jars and all the scoops, and there's no way to get any more samples, so we have to come up, empty the vehicle, and put it back in. So those are the two reasons we might come up. One is just the fluids are leaking, or two, we need more space.
Or we're done and we need to go somewhere else.
We run out of seamount.
Yeah, and so it is on the order of days that we'll stay down typically, like at the max. And it just depends on how tight the vehicle is at the time.
Yeah, we're lucky enough to have enough people on board to operate 24/7.
Yeah, right.
Like efficiently, where we all get sleep.
If we're lucky.
In fact, that's actually the ideal, right? The more the ROV stays down, the more everyone can get into a routine. It's actually when you have to recover the ROV and then redeploy it is when it's a bit more disruptive for everybody, of people having to work strange amounts of hours and extra time.
Yeah.
Bridge nav.
Yeah,
Why didn't I hear you say that?
Oh, I don't know why you didn't hear me say that.
Are you listening on SPL?
You're listening to me.
I was listening to you, and just when you said the move, I didn't hear you.
Yeah.
Oh, that's really strange behavior.
Okay. That's very strange behavior, maybe on my part.
Very strange behavior on part of your ears.
Can you tell me the move again?
015.
015, roger.
Yes, 015. No, yeah. Not 105, 015.
Ooh, exciting.
Thank you.
That is exciting. Yeah.
We all do it. I think I like said 1 because I was like 15, and I was like, no, correct myself.
It's looking pretty gradual.
Yeah.
Rye, how would you feel about doing some flying? Okay.
Hi, thanks Megan Cook. She clarified the longest dive for Hercules, more than 3 days straight in British Columbia in Canada. Nice.
Oh, it must have been ONC.
Yeah.
Yeah, you're just down there futzing with cables and whatnot.
And then we have someone here that mentioned some of the industrial ROVs can go up to a month underwater.
Yeah, that doesn't surprise me.
Yeah.
Like Gabby said, as long as it keeps its fluids inside of it, you just go forever.
And you know how to push the chair back too.
Cool.
Yeah, this one. Yeah. but not too fast because it's moving very, very slow. Adelant's is going to be a hot minute on moving. It's not like when we were at 600 meters. Those were the days.
Gabby and Rai, you guys are not on SPL. Thanks, guys.
Yeah, I'll come on SPL. Okay, that's fine. Okay. Okay. Okay, that's better.
Yeah.
Okay. Did you want me to just take all the comms for you and relay you what you need? Okay. Yeah. I can take the comms for you if it does. That's easy enough.
When do we ever get crazy on SPL?
Never.
Never. Yeah.
The pink is a dead sponge stock with brittle stars on it.
Yeah. This is a great sort of situation to practice hitting your zooms.
Going for zoom.
How many brittle stars can you fit on a stalk?
Oh.
It depends on how long the stalk is.
And how big the brittle stars are.
They gotta be punched.
Are those, what was it you call them, snake-like ones that are really good at interweaving, or is it just any brittle star will just wrap itself around like that?
Usually the snake stars like to be on live corals. And the regular kind brittle stars will do this. So usually the ophiocanthids are the ones that we see hanging out on the dead sponge stalks, but you'll also find those particular brittle stars on like live sponges as well.
How do they get
they're not as picky.
How do they get a benefit from being on a dead sponge stalk?
It gets them up off the bottom and into more turbulent flow. Brings more food to them.
Okay.
Yeah, it's a behavior we see them do a lot. Adelante is on the move.
Does it have any negative impact on the sponge, or is it pretty...
Oh, the sponge is dead, so...
Oh, okay, and they don't go on the living ones?
Sometimes they do, and it could have a positive impact if they're helping the sponge stay clean and clear off sediment from accumulating on a sponge.
Just to give you, it's not a bad thing. It's just to give you a hint really as to like what your feedback looks like.
It's more of a commensal thing where they just live on the sponges, and sponges don't really reap a lot of benefit from having associates.
Because with corals, there's like very tightly coupled associates, right? You get particular species with some corals. Do you ever see anything like that with any of the sponges? Or are the sponges just a physical advantage, opportunity?
Did you see the Regadrella, the tube sponge with the lacy top earlier?
Yep.
Yeah, oftentimes you'll find a pair of shrimps that live inside there.
Okay.
Really nice flying.
They go in there when they're really small, and then they grow up and they can never leave. Asako told us once that it's a wedding gift in Japan.
Oh, really?
Yeah.
Oh, Asako says that the Bolosoma are something that we've seen a lot continuously here, but she thinks that this dive is the first observation from this cruise, from what she knows. So they could be a candidate for collection.
Oh, they collected one of these sponges in 2017 with the Okeanos Explorer from this seamount. Yeah, at the top of the
What
seamount.
a memory.
From this
Holy
seamount.
crap.
Did you transcribe that, Megan, by any chance?
Did I annotate it?
Or annotate it, yeah.
I don't remember if I was the one who annotated that particular dive.
Remember.
But you remember that it was collected.
I remember it was collected.
That's fantastic.
Wow.
They made a video about it. It's
That's
on the
incredible,
NOAA
Megan.
website.
I am totally in awe.
Yeah.
Oh, like I can, if you show me a picture from one of the dives I annotated, I can tell you if I annotated it.
Wow.
Yeah, I have like a really weird memory for these types of things, but someone will tell me their name and I'll forget it immediately. So
Yeah.
you know.
That's really cool. It must have something to do with you enjoying it. Like, you enjoy it because your brain's good at it. Your brain's good at it because you enjoy it.
There's
Yeah,
an
it's a
innate
positive
aptitude
feedback
thing,
loop.
yeah.
Someone's asking what kind of sea star is that? Is that a sea star?
Yeah, those are called brasingids. They are a type of sea star. They are filter feeders. So when they're ready to eat, they'll put their arms in the water, and they have got these little sticky pads, and they'll collect things and bring it to their mouths, which are on the underside of their body. So this one coming up is in the feeding posture.
Okay, so that's with its legs up into the water.
Yep.
And the other one was one who's digesting.
This?
Yeah, the other ones might be napping, you know?
Yep.
What on Atalanta are you looking for?
Right,
you
so
can
then
get,
these other ones are
Argus
just
altitude
digesting
here.
at the moment. Right?
Yeah, they could be.
Okay, and that actually might be a good display for you to have on your right-hand screen. Yeah, that's great. Do that. Yeah, but you did. Yeah, you did. Great. I think those are the two important screens to have. Yeah.
The sponges
Yeah,
that
but you're
we're
getting more
looking at, are
flexible
these
about how
new
you
species
use all the
or?
screens. So that's really
Someone
good.
is asking.
Okay, cool.
If they're one species?
If they're new species.
New species. It was a recently described species.
So it helps to be...
So for the longest time, we were just calling them Bolosomini B. But I believe a paper was recently published describing this species from the sample that was collected in 2017.
Oh, okay, so the first description was from samples from this seamount, or just from this area? Oh, actually from this seamount.
From the seamount.
How cool. So from that perspective, this species is as new as the youngest volcanic lavas that we were seeing in Vailulu.
Yeah, to science at least.
Yeah.
And Megan, is that a Villagorgia?
Romilogorgia, yes.
All right.
And there's a lot of these Volosomas. So many.
It's misbehaving. Let me see if I can play with a zoom and get its brain together. No, go ahead.
Oh yeah, I noticed it doing that last night. Well, you did say you were going to power cycle.
Is the button blue?
Yeah.
Is it just me or I can't hear Gabby?
Sorry.
Okay.
Here we go. I'm back.
Thank you.
Okay, I'll bring these cameras back up.
Yeah, I had a chance to catch up with Matt this morning, and we were discussing these cameras and the extent to which they remain an engineering research project in themselves.
Yeah.
We, I think we got as far as reassuring ourselves we can see photographs from that photogrammetry survey we did at the vents.
Oh, great. Okay.
So we can see them on a hard drive, but Matt is still trying to work out how he's going to access them and turn them into something. So.
Okay.
Oh.
It looks like the lower
That
camera is
looks-
doing better.
Yeah, it's
Yeah,
much better.
let me
That
do
looks
a-
really cool. With all
Oh, yeah, it looks amazing.
That's a great field of view.
Please take that picture.
I've taken that
Look
picture,
at all
and I
those
will.
chromosomes.
I've taken it. I'm going to take it again. It's well worth taking more than once.
Yeah, there's another species
can-
in the same subfamily that for the longest time we were calling the ET sponge because
yeah.
it looks like ET's head. And it's been recently renamed to Advena magnifica, which means magnificent alien. So we don't have to call it ET anymore, but this landscape reminds me of when we were doing a dive up in the Papahanaumokuakea Marine National Monument, and all of the sponges were kind of like hanging out in a group, and they were all facing the ROV. It was like a little unnerving. It's like they're watching us. Yeah, see, there's one of those same brittle stars that we saw on the dead sponge stock, but now it's on this Chrysogorgia. So it's doing the same thing, getting up off the bottom. But we almost always see that association of the little squat lobster Europtychus inside the branches of those Chrysogorgiids. Bridge, Steph.
Yeah, it's amazing.
Can we move 20 meters, 015?
So getting it back online was really a matter of
Thanks.
rebooting the Triclops bottle, not just the camera. So it was the control system for the camera that was all bamboozled, or the computer that was sending data up. Watch your altitude.
Yeah, that's something we haven't seen. Bamboo. Said bamboozled, and I was like, not our bamboo corals. Lots of Chrysogorgias. I've seen at least 3 of these Bathopathes.
Do bamboo corals have a specific depth they like to live at?
They like to live at this depth.
Oh.
On other seamounts we see them, but this one seems to be very sponge dominated instead of coral dominated. There's a lot of these Persingids.
Oh, this is interesting. Asako's volunteered that the wedding gift sponge, it's been discussed as a wedding gift in Japan, but she's never actually known it used as a wedding gift in her sort of like living memory.
Okay.
But it was a custom already 100 years ago.
I see.
And at that time, they would actually be sold as souvenirs in Japan, as things that you could actually present as wedding gifts.
I mean, if I
How
got
did
a
people-
QC sponge as a wedding gift, I'd be pretty happy.
But imagine, like, that was 100 years ago, that was something that people did know about marine biology already. That's cool, isn't it?
How did they collect them?
Did they just wash up on beaches?
They probably dredged them
Oh.
Be like just
from fishing.
fishing,
and stuff.
like bottom
Yeah, yeah.
trawling. It would be like a catch, a thing that came up.
Yes. Sure. Okay. Okay, look at all the cool things. We do have a lot to look at.
I just don't trust biologists at the sticks. They're so
Like, ooh,
flighty.
octopus.
I just don't trust pilots doing the species observations.
Yeah, exactly. No,
It's
the
good, like-
biologists are good at flying. It's the
Yeah,
fact
yeah.
that they're easily distracted by critters.
I mean, if the goal is to be distracted by critters,
The
it's
goal
exactly
is actually, in some cases, to be
That's pretty
distracted
much
by critters.
that's the goal of the mission, right?
Yeah.
But, you know, I guess, like,
But Gabby
doing
would
some
just be spiteful,
engineering-based
and
dives. You know,
Gabby would be referring to the list and say, no, we haven't sampled any black corals. You have to get them.
Oh yeah, you'd
The
be
nemesis
sampling
coral.
so many Bathopathes.
Nemesis gorgia.
Nemesis gorgia.
Nemesis pathes, I think.
Oh, sorry.
Yeah, it's black. Black coral.
The chrysogorgians can be pretty hard, too. They're very...
That's true. They
thin.
always get stuck. We always think we're going to slurp them, and they always get stuck in the slurp.
Yeah, they're just stiff enough that they get stuck, but they're also very flexible.
Can you give us a Doppler reset?
Of course.
Great. Just for reference, I have the camera pointing almost down. No, Atalanta camera. And I'll move it up if you get ahead.
Got a question coming in. How could one start getting into annotating some of the videos? Some of them viewers would love to be able to ID and watch some of these videos. I guess they want to become a Megan.
Okay. So what I would suggest is there is an app for your phone called Fathomverse, and it shows you pictures from dives just like this, and you have to try to identify it. So it's a little game, and you get points for every correct identification you do, and as you learn the animals, they give you, you know, tips and tricks on how to identify all the different types of animals. And as you get further along in the game, you'll start seeing imagery from other dives that maybe have not been identified yet, and that will actually lead into machine learning and increasing our databases. So it's a nice citizen science project where you actually get to be a little bit of an ROV pilot and a little bit of a biologist. And that the game is developed by Embari to help feed into their FathomNet program.
And if you get 100%, you get a phone call and a job offer.
Maybe.
And then you could be one of the 3 Megans that we have. On this
The 3 Megans.
The Megan with 3 brains.
Is
Navigator, pilot, and biologist.
Is there a place that the general public can see the annotated videos, or is that more like
A search.
harder to access?
So all of the videos that I have annotated are all open access, publicly accessible. So all the video that we collect here on the Nautilus can be requested. So you can watch it live right now, or you can request the video from OET at a later time, and you can always rewatch it. Also, the Okeanos Explorer has open access video where they have a data portal online where you can download the video to watch. And some of the videos are still up on YouTube. And the annotations that myself and the team back at the University of Hawaii generate are all available on the NOAA's Deep Sea Coral database, as well as on other databases like OBIS. So you can actually look up the annotations that were made from dives just like this one. And if you're a scientist or a student, or you're just generally interested, you can see what that data looks like. Maybe use it as a project to do some analysis on what types of animals live on seamounts and at what depths.
That sounds great.
Comments here. Annotated
Great,
stuff is
Snap.
on SeaTube, And then live stream oceanographic discovery server is also a great resource, open to the public.
Yeah, there's a number of science, citizen science programs out there. I don't know, I can't like list them off the top of my head, but a short Google search could lead you towards different citizen science opportunities that'll help you, you know, kind of get the sense of what working with this type of video is like. And there's things that are from the ocean, there's also things on land.
I mean, the ocean stuff, though, I mean, it's a rich resource, but also there's lots of opportunity to do new things, right? The amount that we get to do with the resources we have is only so much compared to all the different ways you could use these data and the imaginative ways people could make use of different things, or comparing and contrasting different dives from different regions. So anybody who's listening who has any inclination, you should feel welcome. That it's probably quite easy to find new things to do that nobody's done before with the data we have.
There's a new coral, the little leggy one.
Do you have an inclination what that coral is, Megan, already, as we approach it? Or do we need to zoom first?
Coral, but I definitely want to zoom to confirm. It might be something we might want to collect.
Okay.
Oh wow, look at those polyps. They're like so glassy.
Should we stop the ship? Yeah. Just in case. Yeah.
Bridge nav. Can we hold position? Thank you. Yeah, I want it.
Yeah. Yeah, it's not even a...
Yeah, we might want to come around to the other side of it.
And is this a snip kind of sample or a snip and slurp?
Snip and slurp might work best. Yeah, I think go the other, in that little ditch.
Round to the right of the pink coral. And if we're doing snip and slurp, is this one that's going to be heavy, or is it going to be neutrally buoyant?
It should be a little heavy. It shouldn't be as neutral as, say, a Bathypathes.
Yeah. getting an
Could we
extreme
also
close-up on
zoom
lower.
in? You see that little white, like, star? Yeah, that little white star. Yeah, that's very nice.
Going for zoom.
It's so wee.
Yeah.
Well, whichever data, do we have things in the suction jars?
4, 5, 6, and 7 are free.
Okay.
5's a little sticky though, Gabby.
We can suction it,
Yeah. Yes.
or we could put it in a box.
But yeah, the boxes open are, there's 8 on the starboard biobox, 3 of the small ones are open.
Okay.
Or yeah, 4, 5, 6, and 7 slurps.
Slurp it. Okay. Yeah, it does look pretty thin. I know I'd like a slightly better zoom too. I want to take a look at, the polyps look really strange. They look like they're Oh, that's a weird snail.
You okay with me coming in all the way to look at the polyps?
Yeah.
Okay.
Yeah, they're just like lined up perfectly planar, which, yeah, I think snip and slurp it. Oh, and Chris, just make a note that we should process this one first when you take it into the lab later.
Copy that. Will do. I did. Oh. Yep, that's in the wrong.
Is it alive?
Check 2.
Is it climbed across from 2?
So 3 has sponges. What's in 2? Another sponge?
More sponge.
Oh, you already got a bolosoma. See, we didn't even need another bolosoma.
Wow.
They already got one.
And what's in 1?
Another
Okay.
sponge.
Yeah, that's weird. It says 4 is empty here. So I'll go 5, 6, and 7 are open then. Let me double check this.
So do you want the branch that's got the associate with it as well, Megan?
We could grab that one. The one that has the brittle star on it.
Is it useful to have the associate as well?
There are some people who are very interested in the associates of corals.
Okay.
So
yeah,
they do appreciate associates.
There we go then.
I just think it's really interesting the way these polyps are arranged on the branch, like directly opposite of each other in like this really planar way. I can't remember seeing a bamboo coral like this before.
How well documented are all the different species at this depth? Is it, likely that there's new species that we would see, or is it,
Oh, it's highly likely
Highly
that
likely.
this is new.
Okay.
Yeah.
Cool.
We've done a number of dives at different places. There's been one other dive here, but one dive is not enough to really survey this entire area. And it was a relatively short dive. They were only on bottom for, I think, like 4 or 5 hours. And they just did the very summit. We're not even there yet.
Nice job.
So, this is slightly a new area to be surveyed on this mountain. It'd be like, only surveying a corner of a park and saying, oh yeah, I saw everything here.
Hang in there, little associate.
They do, these ones like to bail. They'll actually like feel the ROV coming and they'll jump off the corals they're on.
I mean, that's smart. I would also do that if there was a thing the size of an ROV coming to me.
It looks like it's trying to bail.
Off.
Yeah,
it's
gone.
There it is.
there you
See?
go.
See you, buddy.
Oh.
It's
Go find
Small
another coral
beers.
to hang out on.
I didn't put its hands under its chin on the life jacket.
That's cool.
These ones don't actually eat corals, but there is bare skeleton. Like it could have gotten damaged, but there are a lot of sea stars that do eat corals. Yeah, as long as we have enough tissue for identification. What will this sample number be?
Be 220.
220.
That handle's determined to get exactly where you can't see the opening.
Totally.
I say it's in. Boop. It's always so satisfying.